Okay, so we've gone over this. If an allele is a version. I know one thing I want to do. Come on. There we go. Oh, yeah, come on. Okay, so smallest to largest.
Largest is base pair.
What?
Largest is base pair.
The way around. Yeah. Okay, so which is the smallest? Okay. Which is the next smallest? Hmm? Yeah, it's a little funny. I probably should want that out. Okay. Next one after that. Amino acid or protein? Amino acid? Protein? Oh, come on. It's 50-50. Just take a guess, okay? Amino acid. Protein. Okay, good. That's good for you, but it's wrong. No, something's got to be wrong. Okay. I'm wrong all the time. I love being wrong because then I learn something new. But sometimes it's expensive to learn something new. Like, the only time you should drink and drive is when you don't have a car. Or not drink and drive is when you have a car. Yeah, that's right. The only time you should drink and drive is when you have a car. There's another one. The only time you should drink and drive is when you're in no condition to walk. Right? Okay. Have you ever been like that? Anybody in here? Oh, come on. I know some of you have. Falling down, crawling on the ground, licking the ground, thinking, oh, it's beautiful. No, that's just vomit. Never mind. Okay. So. And we'll leave chromosome alone. So remember that you have a triplet codon, that means 3 base pairs that code for a specific amino acid, and then So you take 3 of those. You need 3 base pairs for an amino acid. There's 20 amino acids. You string a whole bunch together, and you get a protein. Now, sometimes you can get different proteins with the same sequence because of the way they're folded. So, it used to be thought in the old days that there were something like 1 million different proteins that they had identified in the body. Okay? And they thought that, so each of those probably has a gene that codes for exactly that protein. And then they did the Human Genome Project. Anybody remember that? Where they sequenced the whole human genome. It cost about $3 billion. Took about a lot less time than they thought. And then since then, they've been discovering things. Well, instead of 3 million genes, or 1 million genes for 1 million proteins, they found 20,000 genes. That's orders of magnitude less. So they started thinking, what the hell's going on here? Well, it turns out different genes, depending on how they're expressed, can turn out different proteins just by splicing differently. So I'll give you an example of that. Okay, so we have this gene. Okay, we've got this gene, and okay, and we've got let's see, we've got ACT. GC TA. And then we've also got, so that's the kind of pattern. And so we've got the section that is this. And then we've got a section called an intron. That's a non-coding sequence that gets spliced out. So when you have a sequence like this, you get an RNA, and the intron would get spliced out, and you'd be putting the whole thing back together. But you can actually leave out part of the exome, the part that's actually coding for something, and get a slight variant of the protein. So instead of 1 million genes for 1 million proteins, you can get 20,000 in all sorts of different combinations. Much more simple, and also, obviously, if you had, let's say, 1 million genes, you'd have a pretty big genome. Now, I just want you to know, a bigger genome does not mean that you're better. Corn, domestic corn, has a genome of about 120,000 proteins. So corn's more highly evolved than you are, if you want to take that point of view. So next time you eat corn, you should thank it. Okay? Thank it, yeah. Okay. So here we are reviewing again. Which of the patterns can't belong to a child of the couple? So let's see. A looks like okay, right? Mom up on the top, dad up on the bottom. How about B? Yeah, B's okay too. So here's mom, here's dad, here's dad. Okay, here's mom, here's dad. Let's see. Oh, this is impossible. Why is that impossible? Well, they're both from Dad. Okay? So let's look at this in more detail. What? Oh, you're right. No, B is fine. D. D? Oh, you're right. My mistake. You're right. God. Yeah. No, you're right. Okay. This is okay. You guys are better than me. Okay. Good. Just testing you. Okay, those are fragments. Well, no. One of badge is right between one to the mom, and then one of them is short. Okay, so there is a fragment. All the fragments are moving slowly, and the fragments Okay. Let's see. I just had a good question. I didn't record it. So, remember, these bars represent fragments. Short tandem repeats, right? And the ones that are shorter go farther down. So the one that's shortest of all is that one on the bottom from Dad. And the ones that are longer move more slowly. And so the one from Mom on the top is the longest fragment. So these short tandem repeats can be all sorts of different sizes. But you have 13 different ones, and they can all be different sizes. And they're all going to be a certain size. Okay. Okay. Which cannot be a child of this couple? Oh, okay. One second. I just want to show you something. Okay. This is actually a double band. So it's the same STR and it's landed on the same spot. So they're both the same fragment. Okay, so in other words, this diploid, this guy is diploid. And the only way that can happen is if both Mom and Dad share one standard short tandem repeat. So let's look at this. Okay. So here we are. Is this possible? Is C possible? Okay, look at the top band for Mom and Dad. In fact, they're identical. So you have a double band there. Now let's see. A, we've got Mom and Dad. B doesn't look so good, does it? Okay. Oh no, it's fine. B's fine. Okay, so I thought it was just, remember here, we've got Mom and Mom, but we've also got Dad and Dad. Okay, one or the other, and then we've got this other one from Mom. So this is actually Dad. And this is Mom. Okay? Now, we've got this one, of course, that I already said. It's Mom and Dad. They're identical. And then this one, no, it's a double band. But where is it getting from? Where's mom and where's dad? Well, there's dad there, but there's no corresponding band for mom. Okay? If there was, then you could get that. Does everybody understand that? Maybe not. Who doesn't? I know someone doesn't. Okay. What?
Only D cannot be something.
Yeah. No, it's, let's see, yes, only D. Okay? So, D is a double band at that same length. It shows that the only way that D can be true is if A is the child and the father. In other words, incest is best. Okay? It's the only way that can happen. If, in fact, you had a child that had an allele and mated with a mama, it has to be, and it has to be a female, and mated with the father, the father, so it's actually the mother. The child is the child of the mother and the sister, both at the same time. It's the only way that can happen. Those folks at the FBI wouldn't know what to make of it, because they're stupid. Okay. This is more kind of a review. What kind of cell is this? Okay. A. The only organisms that have a nucleus are eukaryotic cells. And they also have all these other things for making proteins. So you go from making an RNA to a protein. We'll get into that in a little bit. So who are these guys? That's good. Watson and Crick. None. McLemore and Lewis. I don't know who those guys are. Darwin and Wallace. Only if Darwin had known, they would have gotten a lot farther than they did. Darwin was great, but he had no idea about particulate inheritance. In other words, different kinds of different alleles for different genes. And these guys, of course, got a Nobel Prize in 1953. Okay. What's the complementary sequence, complementary base pair to C? Okay, G. What's the one to G? What's this one? What's this one? Good. Excellent. I'm proud of you guys. Okay. Okay, so the double-stranded DNA molecule unwinds. It does that at, as it does that, an enzyme called DNA replicase adds the complementary nucleotides to the single strand. So let's take a look at this. Get it bigger. Okay, we're unwound here, and let's say we'll say So we'll say right here, that's a T. And so right across from it would be an A. Okay? So once you have one strand on one side, you can replicate it on the other by just fitting in all these complementary bases. So where the A is, you have to have a T. If we had had a G, we'd have a mutation there. It would probably be kind of a bad thing unless it didn't code for anything. So, you guys remember, let's say you have a little bit of tissue at the crime scene. In fact, all you have is the butt of a cigarette, and hopefully there's a few cells on there from the criminal. Well, we don't know the criminal actually smoked the cigarette. It could have been the victim, but we can find out. So, we don't have very much DNA. How do we amplify it, make more of it? What is that called? PCR. Polymerase chain reaction. Good. Okay. So here we are on the outside, we have these parental strands, and then on the inside, the ones that are being copied. Now, this happens when either a cell is making a copy of itself. In other words, the cell undergoes something called mitosis, which we'll learn about a little bit later, or when a cell is making gametes. Okay, so, and that's called meiosis. Okay, so why are short tandem repeats like this THEC over and over and over and over again so useful in forensics? Would one short tandem repeat tell us anything? No. But a whole bunch of different ones, then you could easily tell which, you start excluding different individuals because it's very unlikely that any two people are going to have the same short tandem repeats. It's about 1 in a billion. So this is what they do. This one is 1, 2, 3, 4, 5, 6, 7. 7 repeats long. Okay, so remember again, let's talk, hey, over there, hello, hi there. Talking. Okay, whoever's talking, explain this slide to us, okay? Who was talking over here? Come on. Someone was. It'll be good for you. I'm not gonna, this is actually gonna help you understand. Okay, who else wants to explain this slide to me? Okay, stand up. Come on to the front. Come on. No, really, that's fine. You're a smart guy, I know this, so you're going to help us.
Go with him.
What, him instead?
I'll go with him.
Yeah.
I'll go with him. He's the one who knows everything.
One of you guys stand up, I don't care. Okay, come on, let's go. Come on, it's fine. I'm not going to diss on you or anything. How about extra credit? Okay, come on up.
How much extra credit am I getting?
I don't know, probably a couple points anyway.
That sounds good.
Yeah, okay, so here, you can use that. So, you can explain what's going on here.
I really don't know what's going on.
Okay, so we have those fragments up there, right? You see those black lines on the top?
Yeah.
Those are wells. So we take a pipette and put them in wells. Okay? Then what do? It says, read that. Applying
electrical current across the gel negatively charged DNA moves toward the positive electrical.
Right. And then in terms of fragments, which ones go farther down than others? The short ones?
Short ones.
Yeah, good. Okay? DNA fragments move through the gel at different speeds. This is what we just went through. Right? And that's it.
That's it?
Yeah. Let me I'll come up after class and I'll make sure. See, all you guys should have raised your hand. Oh, me, I was talking. Gene. So one long continuous piece, but just so you know, in this gene, oh no, I made a whole lot. That are, and, okay, so a chromosome is one long DNA molecule from one end to the other. Millions of base pairs long. Along that chromosome, there are many different genes. And then it says, how is this one continuous piece of DNA organized into discrete genes? I kind of just told you. But let's take a look here. So we've got this, okay. Here's our gene. What is this adjacent piece? Anything? So we'll say a gene, this could be not a gene. This could be just a bunch of random repeats, let's say, that doesn't code for anything. Now, I want to show you one thing. So what happened to everything? Okay. Oh, there we go. Okay. Okay. Just so you know, banish this from your mind. This is a gene. It's kind of like, that's not a knife. This is a knife. Right? Okay. So, this is the whole gene. Oh, come on. I don't want that. Oh, okay, that's what I did. Okay, I got it. Okay. Now, let's do another one. Transcription. You have DNA. It gets turned into a complementary RNA, which is a single strand that's complementary to the DNA. In the beginning, that RNA strand has all those introns in it, but they get spliced out. Okay, here we go. Here we go. We got G, C, T, G. Then we've got T, 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 T. Then we've got T, A, T. Okay, so this whole thing is a gene. Okay, whole thing is, but the exon, the one that's coding for something, is this. Okay, and this is the intron. and this is another exon. So what actually happens is we get a complementary pair. Also, by the way, this will not be 4, but it'll be some multiplier of 3, because a codon, which is 3 base pairs, codes for an amino acid. Okay, so this will be spliced out, and the final gene will look like this: G, C, T, G, T, A, T, T. Okay? So that's actually what happens. And then further along down the line, there'll be another gene, where there'll be exons, the parts that actually code for something, and introns that don't. Now, there's 2 different hypotheses for why this is happening, why you see these introns. One is that they're mostly junk DNA. In other words, a lot of them are pseudogenes, genes that used to be operational but are not anymore. And they don't get tossed out. They just get kind of put together off to the side and they're not translated. Another is that these introns are actually kind of important. They don't code for anything, but they actually regulate gene expression. Because when you put all these different base pairs together, it's a chemical reaction. And chemical reactions proceed at a certain pace. You have these enzymes that help it get through the reaction. But it might be that, in fact, the longer the intron is, the slower the reaction is, because you actually have to read all the base pairs, and then those are snipped out. And so if you have them all put together, it might not fit together because the reactions are at the wrong speed. The reaction is actually happening. This is something you don't really have to know, but okay. So a gene, it's a structured DNA, and has a recipe, say, for a certain protein, except that you can actually, with one gene, make a bunch of different proteins that are related to each other. That's why there's only 20,000 genes, but millions and millions, over 1 million different proteins in our human body. Okay? So, it's not exactly like a recipe. Sometimes you'll do a recipe where it calls for, let's say, a pound of oysters, right? But actually, you don't have oysters, and you substitute, let's say, shrimp. It'll be a different protein. It might have a similar structure, but it's not identical. So the same gene makes different versions of a protein. So when all this is happening, this replication and everything, all that is done by proteins. Those are the things that actually happen. Proteins that are made by this process are also doing all the work in copying genes and making new proteins. Okay. So we've got, now remember something. We've got a chromosome that's homologous, and on the chromosomes we also have these separate things that code for proteins. And what we do is we unwind those strands from the one chromosome, make a copy of it. And what happens is, there's a codon. Okay. Okay, so we've got a codon. That's 3 base pairs. Yeah, that's right. 3 base pairs. And each base pair, 3 in a row, codes for a specific amino acid. So you go from the DNA to an amino acid. And then you string all these amino acids together, right? And this whole thing, we'll say, is a protein. A protein can be very, very long. Very long. Now, you can change a protein, like I mentioned, in terms of splicing. So you might make a certain protein shorter than another from the same gene because it has to do something slightly different. Okay. So, oh, come on. Here's the DNA again. What I just showed. It unwinds. You synthesize this called messenger RNA, which is single-stranded. The RNA is ferried out of the nucleus and into the rest of the cytoplasm. So, this is the cytoplasm of a cell, so we have a general look at a cell. And here's the nucleus. And all this stuff is happening, and then it's ferried out. And we've got that RNA strand, mRNA, and there's the whole thing is the cytoplasm. These are all amino acids that get strung together. And what happens is that strand is like a template. And it says, okay, this amino acid goes here, this amino acid goes here. It's then from transcribed, it's then to translated. And it's all done through this thing called a ribosome. And a ribosome, there's another kind of RNA in it called translational RNA that then pieces those amino acids together into the full protein. Let's see, I think maybe I'll do a top hat right now. Okay. And then okay. And what is made out of this, hold on. What's made out of this is this RNA. Oh, RNA. Sorry about that. Okay. Okay. We've got this RNA. Now, RNA is called messenger RNA. RNA is always single-stranded. It makes an exact copy with complementary base pairs from one of the DNA strands. Okay? And the strand that it copies, all the introns are taken out, all the amino acids are strung together, and then we'll take a look at